Cool.
Admit all.
Hi, everybody.
Hey, Derek.
Hey, Derek.
Let's go.
Go, Derek.
Woo!
Hi, Eric. Derek, can't wait to hear you talk.
Hi, Derek. Love you.
Hi, Heather. Love you guys too.
Hi, Derek. Good job.
Hi, Derek. We love you in the coconut house.
Hi, Derek. I really like your shirt. It's a very good choice.
Whoa, whoa.
Whoa, whoa. Hi from
Are we ready?
Hi, Derek. We love you.
Go, Derek.
You guys are going to make me blush already. Stop it.
Derek, are we supposed to keep our video on?
Yeah, whatever you want to do.
But we mute ourselves when you talk, right?
Definitely. They'll mute. You'll all be muted.
You want to meet my Kiwi friends?
I have to exit full screen.
Okay, I'd like to welcome everyone to our last scheduled Friday seminar for the fall semester of 2020. At this point, I'm going to turn it over to the chair of the committee, Brian Bowen.
Thank you, Celia. Thank you all for coming out. We're broadcasting live from the library at HIMB, and it's a real pleasure to introduce the dissertation defense of Derek Kraft. Derek got his bachelor's degree at Oregon State University before he volunteered in the HIMB Shark program, began his PhD in 2014. Get that, graduate students? He's out in 6.5 years. He has fulfilled the requirements for the EECB specialty, and in the course of his 6.5 years, garnered a very remarkable $27,000 in grants, or over $27,000. Okay. His professional accomplishments during this period include 11 conference presentations, both on a national and international scale. He won the Best Graduate Student 5-minute talk at Testers. He's been a Sea Grant Fellow, a graduate assistant in the Water Resource Research Center, and was a TA teaching in the biology department. He's been a busy guy. These are publications that have resulted from his work here. I think, including the stuff that's in prep, I think he has 4 first authorships and several more junior authorships, and more coming. He's had additional character building experiences, including learning how to kiteboard. Did you learn that here, Derek? Oh, he learned it at Oregon, but I bet it's more fun here. And he was an essential partner in the Fish Flow project that was led by Mark Hickson. And in particular, he was helping Richard Coleman on his tracking surgeonfish, Menini larvae, documenting where the adults were and where the larvae settled out, which is the arrows on this figure. I don't know how many thousands of hours Derek spent on this, but as he's fond of pointing out, in order to improve conservation prospects for these fish, he speared about 1,500 of them. Okay? Now, Derek is a remarkable case because he actually had a career before he came here. And he decided he wanted to work on sharks. He moved to Oahu with no promise of prospects volunteered in the shark program under Kim Holland and Carl Meyer. And this is the career he came from. He was a head distiller at Rogue Brewery in Oregon. Now, we all know how important it is to promote diversity in our programs. But Rob and I have a little different dimension on diversity, which is that we like to admit graduate students with a diversity of experiences. Like you can ask Michael Hoban about the years he spent in Antarctica, okay? And this was a really different experience. So I showed Rob this application. Hey, look, this guy's a head distiller. Rob said, well, we should take him. And it's not because of the alcohol. It's because of the very different experience, that diversity that Derek brings to our program. And with that, I'd like to introduce conservation genetics of sharks is the topic. It's mostly about silky sharks, but as you'll see later on in the presentation, there's also a clever bit about shark bites. Okay, so please join me in welcoming Derek Kraft.
Yeah.
All right, let's get this switched over. Okay. You guys see me all right? Probably.
You are still in
You're on presentation mode.
Oh, okay, hold on. How's that?
Perfect.
Thanks. Thanks for that introduction, Brian. That was great. All right, let's get into it. So here's a little bit of an outline of my dissertation work I'm going to talk to you guys about. My introduction is kind of a long fisheries management lecture, a little bit about sharks. I'm going to explain to you why it's really important that we look at genetics and how that relates to fisheries. Get into my first chapter, which is a methods comparison looking at traditional mitochondrial markers versus more of a genomic approach and how that relates to outlining stocks of silky sharks. And then I'm going to apply that same method on a larger global data set and talk about all the management implications that research resulted in. And then I get to tell you guys about a really fun side project I got to do out here, which is identifying shark species based on saliva left by bite impressions. And then I'll go through my acknowledgements, which will probably be way longer than it needs to be. So open up a drink and just enjoy. So I imagine most people watching this talk today, I don't have to stress how important our oceans are, not just to us, but to the whole climate. Just climate regulation, it creates the air we breathe, how important it is to the economy, how many jobs are based around things at sea. It supplies a lot of food for a lot of countries and for a lot of people. And of course, there's good recreation. And that picture has nothing to do why I chose this, but our oceans are also under a lot of threats. There's a lot of health stressors. So climate change includes things like temperature rise, ocean acidification, as well as other things. Also, a lot of pollution is going into our oceans. Not just the microplastics that everybody talks about, but we have lots of runoff from industrial agriculture, as well as golf courses, things of that nature. Oil spills we've all heard about. And as well as another thing I wouldn't talk about as much, which is ghost fishing. So we have a lot of derelict fishing gear that's floating around our oceans, continuing to catch fish, though we'll never see them. And the big topic that I'm going to focus more on today is overfishing, which is happening in almost all of our oceans at all sorts of depths. So to give you guys an overview, the amount of fish we extract from the ocean is a lot. So This is a figure from 2010. It's showing a slight decline in fish catch, which has kind of leveled out even over the last 10 years. And it lands us at around 190 million US tons. This number does not include illegal, unreported, or unregulated fishing, which is about a third of the global catch. And that little bit they have up there for discard, I also think is a gross underestimate. But since these are the numbers we have, let's run with this number. What is 109 million US tons? So the LA Coliseum, okay, it can hold a volume of about 2 million metric tons, okay? So I gave you US tons, let's convert that to metric. So 109 million US tons is about 79 million metric tons. So if we do the math on that, every year we catch somewhere around 40 LA Coliseums worth of fish from our ocean. That's what we harvest from the ocean. If we break that down a little bit further, that's every 1.3 weeks we fill that Coliseum, and that's from the football field all the way up to the top of the nosebleeds. That's how much just sheer volume we're extracting from our oceans, which is a lot. We don't have a good habit of doing it very sustainably. As we industrialize fishing practices, we're getting better and better at catching the fewer number of fish that are left in the ocean. So here's just an example of some Pacific bluefin tuna. You can see in the last 30 years, they've had a steep decline. And since then, I think it's probably about the same. And as the ocean's starting to run out of fish, we're developing more and more technology to catch them. So this is a picture of what are called sonar fads. And a fad is a fish aggregating device. So there's this phenomenon where fish are gonna gather underneath floating objects, whether they're a palm or a log or a buoy that somebody sets out. And as the fish aggregate underneath of them, these sonar fads then they ping back to the boat how much biomass is floating underneath of them. So the boats can just go around and pick up the fads that have big schools of fish under them. Additionally, in case you can see what's going on there, they're also sitting on the helipad of this boat. So these boats also have helicopters that fly around and can chase down the free swimming schools of fish, alert the boats where they are, and they can go catch them. So this is happening every day. This is some pictures from some fish markets. We're catching a lot of fish all of the time. And the sheer volume that comes out is a lot. And the same goes for sharks. We do have quite a few targeted shark fisheries around the world. Some do bring them in and eat them for meat. In this picture, so some silky sharks are down in the bottom right there. But a lot of them also get caught for their fins, which can sell from anywhere from $200 to $500 a pound. So when you're looking at these tables of shark fins, you can see the incentive to go out, catch sharks just for their fins. But a bigger issue with sharks is a lot more of them are caught as bycatch in these other fisheries. So the long-lining tuna fisheries and obviously all of the big net fisheries as well. The amount of sharks that they catch is also really, really alarming. And when we think about the biology of sharks and how they compare to fishes, we should be extra concerned about how many are being caught in the fisheries. So if we take kind of the average tuna species as an example, tuna are able to reproduce after they're somewhere between 2 and 3 years old. And when they do, the females can produce from 4 to 8 million eggs. The larger bluefin tuna can spawn up to 30 million eggs. And they can do this every 2 to 5 years. So even when these fisheries are hit super hard, they still do have the ability to bounce back if fishing pressure is relieved for a while, like we can see around 1992 here. Sharks, on the other hand, are kind of a different story. So it can take them between 7 and 10 years before they can even reproduce. And when they do, it's only between 2 and 10 pups, some at most in the 15 to 20 range, but not all of those make it even to be adults. And they can only do this every 1 to 2 years. So the rate that they can reproduce is drastically lower than the fishes. I couldn't find a graph of that, so I made my own, so that's made up. But the point is that they cannot reproduce nearly as quickly as the fish can, even though they're caught almost as much. And that's really alarming when we start to look at where shark numbers are at nowadays. So this is a graph showing how percentages of shark populations left since the 1970s, so only in 50 years. The little red sharks on the bottom of each bar represent how many are left. Okay, so it's not very many. And I couldn't actually find super good sources from this graph, but I do know for most of these species, the amount of decline is, and a lot of them are over 90%. So that's just in the last 50 years. We're really losing our sharks out there, and that's an issue. Sharks play a really important role in the ecosystem. Most of them are either top predators or even mesopredators, so in the top 2 tiers of the food chain, and that regulates the ecosystems that they're involved in. Okay, it keeps the lower trophic levels in check and creates really healthy biodiverse reefs. This is also true out in the pelagic realm. So out in the blue water, the bigger sharks regulate, all of the tunas and the smaller fishes all the way down. So if we want to ensure the health of our oceans, it's really important to keep these top predators in place. And so that's where fisheries management can come in and actually do something to try to help maintain these shark populations. They can do things like set catch limits and retention bans. So that means certain species you're not even allowed to bring onto the boat, and gear restrictions that might be a little bit more selective for the target species and catch less bycatch. They can also, of course, restrict seasons, restrict fishing grounds, and set up marine protected areas. And they set up all these rules and regulations based on fisheries stocks, okay, which is kind of like groups of fishes. And I'm going to get into that for a little bit because the stock concept here is really important. Okay, it's basically a unit of fish. Okay, it's a bunch of individuals that are in a group, and they're generally reproductively isolated from one another, which means we would be able to see that in their genetics. So you would manage these 2 stocks differently because they have different reproductive outputs, they're in different places. But the actual stock concept itself is really complicated. It has to do a lot with data availability. We don't always know where these stocks are, and of course, resources to even investigate where these stocks might be. But if we really want long-term management, okay, conservation, if you look up in the left-hand corner, what we really want to focus on are genetic stocks. Okay, so that's where I come in with my research. And before we get into that yet, I want to explain the genetic concept a little better. So let's say in the Pacific Ocean, we have one big genetic stock of sharks. Okay, and all these sharks swim over big areas all over the ocean. They reproduce with one another, spreading their genes around, and therefore all of their DNA is going to kind of look the same based on this yellow color. And then if we have some fishing pressure, say down in the South Pacific, which we have plenty of, and they take out a bunch of sharks down there, because of the nature of these sharks, how they move around the whole oceans, other sharks will eventually make it into that area, and you still have a large population that's able to reproduce, and they'll be all right. But let's say that's not the case. Let's say we have multiple stocks, multiple genetic stocks. So you have these 3 different groups of sharks now. They only intermingle with each other and don't go between groups. Therefore, their genetics are going to start to look different. Now, if we apply that same fishing pressure, we don't have other sharks coming back down to that area. And so these smaller populations are a little bit more vulnerable. And even if it's not a total wipeout, we have less fishing pressure. Those populations are still now so much drastically smaller, they're going to reproduce more slowly. So smaller populations are more susceptible to fishing pressure. Okay, fisheries lecture over. Enter the star of the show. Here are some silky sharks, stage left. Okay, they're awesome little sharks. I hope that's not coming in too chunky for you guys, but they're very good at interacting with fisheries. Here are some harassing some tuna fishermen in the Maldives. So fishermen aren't a big fan of them. One gets poked here in just a second. Bad shark, although he's fine. So yeah, these sharks are involved in fisheries often, and a lot of it has to do with where they are. So the silky shark is a circumtropical species. This is a map of known catch locations. Yellow, where they've been caught some, to a gradient of red, where they've been caught more often. And as we can see, their habitat very much overlaps with the commercial tuna fisheries. And so they're very heavily caught as bycatch. In fact, in some of the purse seine tune fisheries, juvenile silky sharks make up to 90% of the shark bycatch, which is a lot. And their numbers have drastically declined due to this really heavy amount of bycatch. And they're also one of the most prevalent species in the shark fin trade. So with those two big industries combined, these guys are actually the second most caught shark on our planet. When I first started giving these talks about 5 years ago, I will admit, they were listed as nearly threatened, but they've been relisted now as vulnerable by IUCN Red List. The next listing is endangered, so they're certainly a species of concern. Currently, silky sharks, along with a lot of other species as well, are managed by these regional fisheries management organizations outlined in these colors here on this map. So there's only four main ones. The one down in green at the bottom, they're a little too far south for silky sharks, but currently they're managed as one stock per RFMO. So what that assumes is over in the Atlantic Ocean, it's one big group of sharks all swimming around together. In the Pacific, you have one on each side. They know not to cross the line there. And then over in the Indian Ocean, it's one big population. But I really want to know, does this management scheme reflect the biology of these sharks? Is this adequate? Are we managing for the biology, or is it just convenience, which of course it's definitely out of convenience, but anyways, getting away from that. So there's been a couple genetic studies done with silky sharks before. They've all focused on just a small portion of the mitochondrial genome. Here's one from 2013. They looked across the Pacific Ocean and they found weak but significant structure from the west to the east. There was another paper done in 2015 where they looked over a lot more areas of the world and they found some structure between the Atlantic and the Indo-Pacific. And these Gulf of Mexico and North Atlantic samples I'm highlighting because they were present in the next study as well. So the same samples were used there. And Rodrigo Dominguez did a bunch of sampling down off of Brazil and found a little bit of structure between the North and South Atlantic. So for my first chapter, what I wanted to do was examine these mitochondrial markers and see how they compare to more of a genomics approach. And some of you might not understand what that means yet, but we're gonna get there. So traditionally, we've used single marker genetics, which is looking at a small part of the mitochondrial genome. So here's a schematic of a silky shark mitochondrial genome. And what a lot of people look at is what's called the control region, which is a little bit over 1,000 base pairs. And now, when we look at DNA, hey, look, here's some DNA, finally, right? Okay, so this is what DNA looks like on our computer screen sometimes. Each row can represent an individual, for example. And what we do is we look for the differences. So the single nucleotide polymorphisms, or SNPs. And if we zoom out just a little bit, we can stack a bunch of these up together and see how they compare. And we'll have areas like this blue dotted line here. That's a really nice SNP. We can see about half of the population has a certain nucleotide there, half does not. And we also see areas where a population might have a fully different SNP in one location compared to another. So that's the kind of information we use to compare these populations. So that method's been the gold standard for a long time. You get somewhere between 30 and 50 SNPs when you look at that little region. But now a genomics approach, okay? So this is gonna look at a lot more of the genome than just that little bit. So here's, again, the mitochondrial control region circled in blue, but with the data set we collected, we were able to look at the entire mitochondrial genome, okay? So all 17,000 base pairs. And on top of that, we were also able to look at lots of nuclear DNA, so all of the other chromosomes that have a lot of information in them. And for these Carcharinus sharks, which is the genus of sharks, there's about 70 to 80. So there's a lot of information out there. And the mitochondrial genome and the nuclear genome are inherited a little bit differently. So we actually analyze these two data sets separately. Okay, so the breakdown is for genomics, we're gonna get a lot more markers. We're gonna have the nuclear and the mitochondrial data set, and there's a lot more biological information, which is a lot like drinking from a fire hose of information. So to do a direct comparison, we use the exact same samples as this 2015 study. So the same Gulf of Mexico, North Atlantic, and Brazil, just to examine the Atlantic, and then one outlier group, which was the Red Sea. So here's a map of the samples we used. The type of genomics that we did is called pooled EasyRad high throughput sequencing, also known as pooled seq. And basically what that means is we take equal molar mass of DNA from each of our individuals, and we sequence them together as a single sample. And that brings the cost down a lot. And it's the only reason we were even able to do this genomics work is by using this pooled EasyRad approach. And of course, we just analyzed the data, which was a lot more involved than we thought. So I'm going to talk just a little bit about the analysis. You guys will see my really cool nerd emoji there. When you see that on the screen, if you're having a lot of trouble following, that's okay. I'm going to sum it up afterwards. But for those of you who are kind of into the genetics world, then this is for you. So we used Dedocent to do a de novo assembly to create the nuclear genome, to map all of our reads to it, and then to call SNPs. But after that, we didn't quite have the software we needed to analyze. And so for a long time, I sat in a small computer room with Evan Barba and Emily Conklin and came up with this program called AssessPool, which is essentially takes the SNPs that were called from the data set, does a lot of math with them, gives us our population genetics outputs, and puts them into these really nice figures for visualizing the data. So we can actually explore the data a little bit and come up with our final results from there. And so for that, I'm going to get into them. So results are in the format of FST values. And FST is just a proxy for how different are the genetics between these two populations, okay? It's from 0 to 1, with 0 being there is no structure. So those two those two regions are the same population or the same stock. With a gradient to 1, and the colors here are what's important. So less structure will be shown in yellow to orange with a gradient all the way up to red. And that scale will change per diagram that I show you. And so they're listed in these matrix here, so you can look and compare two different populations and see how closely related they are. So I'm going to slowly fill this in as we go. My results, the genomics, are going to be on the left, and then the results from the mitochondrial control region from the Clark paper will be on the right. So first, let's just look at our mitochondrial genome. We can see any of the Atlantic populations compared to the Red Sea have really high FST values. Anybody that works in FST, 0.6, 0.7 is drastically different. But even when we look at the comparisons within the Atlantic, they do have positive values and they are all significant. So the mitochondrial data shows structure even within the Atlantic. And our nuclear data, shown in the blue at the top there, shows a similar pattern. We have all positive values, which are all also statistically significant. So the two data sets kind of match there. So what that can conclude is we do find structure within the Atlantic with both the nuclear and mitochondrial data sets. And when we compare that to the Clark study, significant values are shown in bold. So the Red Sea did have significant differences between the Atlantic, but none of the significance values for within Atlantic comparisons is significant. So they did not find structure. So what we can conclude from that is the genomics has a lot more power to detect this structure, and that there are more than one stocks within the Atlantic. And what that means for management is in the next chapter. So we take that exact same method, and now I'm going to apply it to a larger data set in just a second. All right, I'm back. Okay, so thanks to Melanie Hutchinson, she organized a boatload of scientific observers and fishermen to collect samples. We were able to get samples from all over. We were able to add one region to our previous ones from the Atlantic off the tip of Africa over there, 5 regions across the Pacific, as well as the Indian Ocean and the Red Sea. And we'll take that same genomics method and now just apply it to all these populations. So the results are going to look really similar. The only difference now is that these matrices will be mirrored on top and on bottom because there's too many populations to try to complicate the data. Okay, first things first. This is our mitochondrial genome for our global assessment. And a pattern sticks out really vibrantly right away, and that is any comparison between the Atlantic and the Indo-Pacific has really high FST values again, like we saw in the first chapter. And the Northwest Atlantic, however, has this kind of intermediate pattern where the FST values are still really high, but somewhere in the middle of the other comparisons, which is interesting. Now, it may look like this is just kind of a flat yellow in the middle of these circles within the Atlantic and within the Indo-Pacific, but that's just because the scale is really off skew with how different the between ocean comparisons are. So I'm gonna pop those two boxes out and rescale our reference there. Okay, so if we look at the Atlantic mitochondrial comparisons, we see, again, the Northwest Atlantic is quite different, or has higher FST values when comparing the other populations. And even within the Atlantic, those comparisons are all still pretty high. And in the Indo-Pacific, we can see the Eastern Pacific and the Indian Ocean are shown to be quite different than the rest of the populations, which makes sense geographically. And then the other ones, however, they are still significantly different from each other. The FST values are lower. And I know the Red Sea is grouped in there as well. I'm going to come back to that in just a second. Looking at our nuclear data set, the pattern is still there, but it's a little bit more complicated because we looked at a lot more SNPs than in just the mitochondrial data set. But we still see some similar patterns. The Eastern Pacific is standing out, and the Indian Ocean is standing out as being quite different than the rest. And even the South Central Pacific is showing higher FST values. We still see the Northwest Atlantic being a little bit in the middle, especially with the inter-Atlantic populations. So things are mildly working out geographically, except of course for the Red Sea, which was kind of puzzling for a while. So the Red Sea compared just to the Indian Ocean, we see pretty high FST values. But when we compare the Red Sea to the Western Pacific, in both the nuclear and the mitochondrial data set, the lowest FST value was between the Red Sea and the North Central Pacific, which is very puzzling. We've tried to discuss this a few times, and the only thing we can come up with is that they have dug a tunnel underneath Asia and are commuting that way, but of course that's not true, that's a joke. What's interesting though is that 2015 paper looking at just the mitochondrial genomes, or mitochondrial control region, they also sampled the Lion Islands and the Red Sea, which is a completely different set of samples than what we used. And they found no differences between these two populations, or between these two regions. But when they looked at the Ascension Islands and the Red Sea, they did find differences. So both studies picked up on this pattern, and that's kind of interesting, and I'd love to hear your ideas on why that is. To sum it up, if you look at both these data sets put together, the major patterns do match each other, and we can test that with a Mantel test, which we did, and it had really high correlation, which of course was significant. So both these data sets, again, are showing us a similar picture, which makes us believe in them just a little bit more. So what does that mean for the global stock structure, right? So what all this data, all these colors, if silky sharks had similar genomes in all regions, represented by these colors here, they'd be one stock across the globe, kind of like the wahoo or the basking shark or true pelagic swimming around large areas. Or maybe it'd be like the whale shark, where you have an Atlantic population and an Indo-Pacific population. But that's not what we're seeing with silky sharks. Every region tested in this study was significantly different from the others, meaning we have a lot more small isolated populations than we previously thought. And when we start to consider some other fields of study, looking at how much silky sharks are really moving around the oceans, we can look at tracking and tagging data. This was given to us by Melanie, tagged by Jamie Barlow. And this is an adult male silky shark tagged around Oahu, and this is over 7 months. It didn't go a terribly far distance. And of course, this is just one example. I'm excited to see the rest of their data when it comes out, when they finish this project, and see if there's any moving any farther. But all of the other literature out there on tagging and tagging and tracking data, we don't see them going super long distances. We don't see them moving across oceans like we do with the whale shark, which twice we've had a tag on when they've swam all the way across the Pacific. So we have this pelagic shark. It's definitely out in pelagic waters, but it has more of a coastal shark's genetics pattern. And when we think about this in terms of management, we'll bring back our management map here of our RFMOs. We can see that there's multiple populations within a single RFMO, which are currently managed as a single one. So within the Atlantic, we have at least 4 populations. Within the Western Central Pacific, we have at least 4. And the Indian Ocean and the Red Sea are definitely different from one another. So as I concluded from earlier, these smaller stocks can be a lot more vulnerable to overfishing. So there's some management suggestions I can make. They need to be managed as separate populations. And managers don't necessarily want to hear things like this from us genetics folks, but you'd have to do multiple stock assessments, and you might need greater collaboration between local agencies to manage these smaller groups, which does create a lot more work, but that's what the data tells us. So as I was working on all of this, I also had a great opportunity to work on a really fun side project with Carl Meyer. For this talk, it's titled Dude Who Bit My Board. And essentially, we just wanted to see if we get DNA off of shark bites. So in Hawaii, we've had 120 human-shark interactions since 1995. We like to call them human-shark interactions because shark attack makes it sound like sharks are very aggressive and malicious, which they're not. These human-shark interactions have resulted in some fatalities, which is awful. But in a lot of these cases, in about, 30 of them, it's been just a surfboard or a sup, so nobody was injured. And of these 120 in Hawaii, 63 have been the tiger shark, which is a little bit expected. 15 were reef sharks. 2 were the cute cookie cutter shark. That was both of those were from people swimming between Molokai and Oahu. But about 40 remain unknown. So a third of these human-shark interactions, we don't even know which sharks were involved. And if we scale back and look at that on a global scale, just from 2010 to 2019, okay, when we have lots of communication going on in the world, information travels really fast, we still have 70% of these interactions the species are unknown. So that's a huge gap in the data. So if I'm a detective and I want to know who might have been involved in these human-shark interactions, if I'm examining evidence like this, what I'd like is it to just be covered in DNA. We just hope that there's DNA all over everything because that makes our job a lot easier. So the essential questions here are, is there DNA on it? And can we use it to see who bit it? The idea being, when a sharks bite onto something, that their saliva is actually pretty sticky. When you think about how they breathe, okay, they're taking in water through their mouth all the time and it's going out their gills. If they had really slippery spit, it would just be washed out all the time. So it has to be really sticky, otherwise they'd have to keep reproducing it. So we're gonna test that theory. Here's intern Kaylee collecting a sample for us. This is a large tiger shark. Okay, here you can see the transfer of DNA. Probably right in there somewhere.
That was one of the most dramatic things that's ever happened on this boat. Intern, give me a Campari, will you? On the rocks?
That last part's super important because Kaylee's an intern and she's awesome. So from there, we bring the board back up onto the boat. We use these polyester swabs. Any of you lucky enough to have had a COVID test have had these shoved up your nose. They're virtually the same thing. And we swab for the saliva on the board. And then we do what's called DNA barcoding. So we have our sample, we extract the DNA, We amplify that target region, okay? So this is that same single marker genetics that I talked about earlier in my talk. And then we sequence it. We get the sequence back, and then we put it into an online database that has tons of DNA matching to species names, and we hopefully get a match from our sequence. So in this case, it would be the jaguar shark. And for our example, with our tiger shark, we ran through this process, we put it into the database, and boom, we got tiger shark back. We're like, wow, that's awesome. We're really excited. This worked once. So we definitely wanted to increase our sample size. We wanted to see if multiple species also worked. Maybe it's just the tiger shark that worked. And also we were curious if this would work on more than just surfboards. So we set out to sample around Oahu. We had our one tiger shark from Kaneohe Bay over there, and then we got 2 Galapagos sharks up on North Shore, and then COVID-19 hit and all of our fieldwork operations were shut down. So we have that many samples from Hawaii. But luckily, I gave kind of a drunk version of this talk in a hotel room at a shark conference last year, and an awesome young scientist named Lauren Meyer said, oh, well, down in Australia, we test shark deterrents all the time, so we have sharks biting stuff. Do you want me to try to get some samples from you or for you? Absolutely, Lauren, that would be fantastic. So here's a white shark biting onto some wetsuit neoprene. Even his DNA all over it, which is great. And with their help, they work over in New Caledonia and the Norfolk Islands and in Neptune Islands. We were actually able to collect 5 bull shark bite samples, 3 dusky shark samples, 7 additional tiger shark samples, and 17 white shark samples. And these were both on surfboards and on that wetsuit neoprene. So they'd pull that out and they'd swab the bite marks, and you do each bite, you would swab 3 times just to ensure you got DNA on at least one of them. They shipped them back to us here in Hawaii, and we did all of our fancy lab work. I had a lot of help from Marianne. And here's just an image of our gel extractions. And I just wanted to show that we get varying amounts of DNA per swab. So each bite is outlined with those blue dotted lines. So 35, 36, 37 are all from the same bite. But as you can see, different swabs yield different amounts of DNA. And even though some of them don't have that much on there, most all of these in this photo worked. I think 40 didn't work, but even 38 and 39 was enough DNA, not to spoil the answer. So this is what our sequences actually look like for those of you that have never got to look at actual Sanger sequencing data before. It's these fun, colorful rainbow lines, and each peak represents a nucleotide. So that's what sequences look like. So of all of our 32 samples that we had, we were able to successfully get DNA off of 31 of them. We had 18 bites that worked for all 3 swabs. So all 3 swabs were able to detect DNA, or were able to get good DNA off of. 9 worked for only 2, 4 worked for 1, and 1 white shark sample on some neoprene, no matter how many times I tried, couldn't get it to work. But 31 out of 32 we thought was pretty good. And you might notice it says successful DNA sequencing. It does not say successful identification, and I'm gonna tell you why. So I was doing our dusky shark samples and ran it through the online database, and this is what popped out. Dusky shark, Galapagos shark, dusky shark, Galapagos shark. And at first I was like, wait, this sample was definitely dusky shark. That's what it says on the data sheet, and I thought I had screwed up. And then I was like, wait, it says both. And it says 100% chorea, as in like, it's a really good match for both. Well, that doesn't make a lot of sense. So I did some homework and I found a paper that actually said, historically, these 2 species were hybridizing. Like, oh, okay. And I found a more recent paper that says they're continuing to hybridize. This is pretty rare in the shark world. I can only think of 3 hybrids with sharks. Looking at Brian. Yeah, so it doesn't happen very often. We happen to capture 2 species that do. So the dusky shark and the Galapagos shark, although their DNA was flawless, I couldn't actually tell them apart, so I couldn't positively ID them. If we did a different genetic marker, we could tell them apart, but for what we did here, we couldn't tell them apart. all of the other species matched their field IDs and we could successfully ID them. So, is there DNA on it and can we use it to see who bit it? The answer is definitely yes, with this tiny asterisk of unless they've hybridized and the marker you used was incorrect and you can't tell them apart, which is very rare. And with our success rate, 31 out of 32, this suggests this could be a pretty viable tool for real life situations. So Carl put together these awesome University of Hawaii shark bite forensic kits. I had one in my car for a long time. I was super proud. We're like ready just in case, not hoping for a real human shark interaction. We staged a few of these at some lifeguard stations. So if somebody's board or leash or part of their clothing got bit, but no person was bit, we'd be ready to real world test this tool. And the reason we can't do bites off of actual people is because the amount of paperwork for bloodborne pathogens, the legal paperwork of somebody's injured and now you're touching them, just no, we want nothing to do with that. However, I do think it would work. So we've actually gotten this to work on fish. We had some captive sharks here on Coconut and we bought some mullet one day and fed half of them to them and swabbed off of the fish. And we were able to get that to work. So if it works on fish, it could work also on human victims. But this now also is an awesome tool to examine things like shark depredation, okay? Where sharks will come in and actually take fish or parts of fish off of fishermen's lines. This is a bunch of photos down from Guam and Saipan where their shark depredation issues are really bad and they were complaining about it for a long time. And we got some funding to actually put together a project to try to characterize this a little bit. So thanks to some awesome folks who helped me put together about 30 of these shark swab kits. Then Carl and I went down to the Marianas Islands. We met with a bunch of fishermen, handed out these kits, did some trainings, and have now started a citizen science project down there, or I like to call it a fisherman science project because our fishermen down there are great. They even let me come on the boats with them in case they got sharked that day and I could collect some samples. I was supposed to go back a bunch of times, but because of COVID, I couldn't. So this is an ongoing project that I'm really excited to see how it turns out. And these really nice gentlemen who took me out on their 20-foot boat are still fishing on days like these, even though they had both told me about all of their grandchildren. And I mean, that was fine, but it was a little terrifying. But I'm really happy we got all those kits handed out. This is one of our fishermen down in Guam, James. He's been a great participant. You can see how happy there he is about his catch. But in the midst of fishing, James takes the time to swab his fish for us. We appreciate James for all his work. Yeah, and that project's ongoing. I'm super excited to see what the results bring. Overall conclusion from all this, genomics can be really helpful as a fisheries management tool in outlining stocks. There's a lot more populations of silky sharks out there than we previously thought. We could probably try to think about that management scheme that results from that. And sharks have some really sticky spit, and we can use it to figure out who they are. If you guys enjoyed any part of my talk today and you want to do what you can to help sharks, think about your seafood intake. If you have the option to not eat seafood, that's wonderful. Some people really depend on it for protein. I understand that. If you are going to eat seafood, you can eat it responsibly. So Monterey Bay Aquarium has a seafood watch. They also have an app for that, so you can check your food before you eat it. And this slide's for you, Brian. Never eat shrimp or canned tuna, because that tuna is most likely not sustainably caught, and they're also going to be catching and killing a lot of sharks. It's something Brian ate in our meeting on Wednesday, so I told him I was going to give him a bunch of crap. Being a fish man himself, maybe tuna at Subway is not the best idea. Whew. And with that, I'm going to thank some people for a little bit. I got to thank Sea Grant first and foremost. They brought a lot of money into my projects with my project development grant. They also funded me through a lot of projects. Maya, you've been freaking awesome. Also getting me set up with my new fellowship. I'll be continuing to work with Sea Grant. I'm so excited about that. The American Elastomeric Society for some of their funding opportunities. Save Our Seas, put a lot of money into these projects. The Hawaii Institute of Marine Biology Lord Scholarship also gave us a lot of money, and I was also on NSF funding for a little while there, so thanks to all of our funders. My committee, Brian, I'll get to you in a minute. Jeff Drazen. Jeff, are you there? What kind of fish is this? I couldn't figure it out.
That's a giant rat tail.
Awesome. Thank you. So Jeff's pelagic ecology class was my favorite class I ever took. Jeff's really great as a committee member. He keeps you honest. He keeps you on time. He checks in, which is great. Zach Forsman, you know, basically he's been a room away from me during all of my analyses and such a big help. Kim Holland, it's awesome having a shark hand like you on my committee and keeping me honest about things I try to claim about my genetics. I really appreciate that. The late Steve Carl, he also was always keeping me very honest. I hope none of this shames him. And Bob Thompson, thanks for showing up to all my lab meetings and defending me when I wanted to drop my shark fin project, because I would have been here for another 3 years. Okay, and a very important person. Just a second. Is one of your guys' mics on? Like getting reverb. There we go. Thank you. Melanie, thank you so much. So I was an intern in the Shark Lab. Melanie was running a boat to get some supplies. She's like, hey, you want to help me move some stuff? And of course, I was an intern, so the answer is yes, of course. And on that boat ride, we got to know each other. I told her I was brand new to the Tobo lab, and she goes, oh, I might have a project for you. So Melanie, right out of the gate, handed me about 1,600 silky shark samples from all over the place. Stuff she put blood, sweat, and tears into collecting, and then just gave them to me. What she's doing now is so awesome, Melanie. I always look up to you. And this is how awesome she is. She actually got her face on a beer can with a tiger shark on it. It's got a tracker on it. She does such awesome work that they wanted to recognize her. So Melanie, you keep doing awesome things. I'm gonna keep trying to be half as cool as you are. The people on the left here are people Melanie organized to collect even more silky shark samples. So there's been a lot over the years. And then while we were doing this sort of thing, all the people who helped with my shark saliva bites are listed on the right there. So thank you to you all for making this project possible. All right, these two. Evan and Emily, not only are you guys really good friends, I mean, Emily, you were my roommate for a long time, but the three of us were kind of in the trenches there for 2.5, 3 years. We all sat in this tiny little room. Sorry, Evan, you weren't actually in this picture, so there you are. Evan and Emily developed a lot of this software that we ended up using. So they would sit there and build it all, and I would sit there and just break the crap out of it and try to use it. And like, hey, this didn't work. Hey, this didn't work. So I was definitely a part of the team. So you guys, thank you so much. Somebody maybe check their mic. It might be on. Thanks. We also had this amazing ability to wear the same shirt on the same day. Evan and I were very good at it, and it brought us joy, so we took pictures of it. When it happened with Emily, she hated it. But we did get this one photo from Japan of it, and the rest of the photos mysteriously disappeared. But just thank you guys so much. You're a part of the lab that's just so core to me. Which brings me to the rest of my lab. From the old guard that was here when I first arrived, and everybody welcomed me with open arms, I felt a part of the family. To the new guard who's here now, and I'm gonna try to do this smoothly, but they knew they weren't gonna be here, so they hung up photos of themselves so I could present to all of them today. Paulo, I know you hung that up, but to the rest of you that participated, I can't tell you how much that meant to me walking in. Thank you so much. There's that same photo. Yeah, to the new group that's gonna be inheriting this dynasty. You know, from everything from us just goofing off at our holiday parties to looking super fancy at the tester symposium when we're cleaning up awards. I mean, you guys are such a family in the best of times, and even in the worst of times, when we go through tragedy together, we still all get together on the beach, and we're there for each other and have a good time. And Paulo, I expect to dive 5 as soon as I see you. And the awesomeness that our lab is brought on by these two. Rob and Brian are our leaders, and they instill such a sense of community in our lab that's just undeniable. Not biased when saying we've got the best lab on island, but we do. Brian has been an amazing advisor. He always is promoting his students. He's always introducing us to people at conferences. I remember my first conference was the Indo-Pacific Fish Conference. It was in Tahiti in 2017. Brian got on stage in that tie-dye shirt and hat and no shoes, I remember, and gave his talk in front of a huge symposium. But then Brian did something I've never seen any other advisor do. At the end of his talk, he plugged all of his students' talks, told them what they were about, what time they were, and that they should be there to go watch. Something I'll never forget, and I've also never seen any other advisor do that for their students. Brian's also the kind of advisor that'll show up to your 27th birthday and every other birthday that I invited him to from then on out. Brian walks in the door with a bottle of tequila, says happy birthday, it's got a ribbon on it. He then sits by the door leading to the backyard, and every single person that walks by, he poured them some tequila and said, "Hi, I'm Brian, I'm the fish guy." He was the life of the party, and in his defense, he has not passed out on my couch. We really wanted Rob to come over, so he, like Superman, across my living room, landed on the couch like that and said, "Send Rob this picture and tell him I need help, then he'll show up." And he didn't, but. Which brings me to Rob. Now, Rob, I offered Rob to be on my committee, and he politely turned me down. He said, "Well, you're going to have my help anyways. You don't want to wait for me to show up to committee meetings," because he's on 30 some odd committees. However, he still makes the time for you, and he knows the ins and outs of publishing and the genomics. The amount of hands he has his pot in is incredible, and he still makes you feel important. So thank you, Rob, so much for being a part of all of this. Another amazing mentor to me is Judy Lemus. So I spent 2 years working in an educational program with her where we brought in science teachers and did projects with them. And that was such a big development for me. And I'm really thankful for the other people I worked with. But Judy especially, not only did she give me career advice, also a lot of personal advice. And Judy, you're just awesome. And I wouldn't be the person I am today without having you around. So thank you. Okay, I also got to thank the Shark Lab. Again, with the old guard of Danny and Mark and Melanie. Mark finally agreed to let me be an intern after I asked him, I think 2 or 3 times over email. And I think the answer was basically, "Oh my gosh, fine, you can show up." And that's why I moved to Hawaii. So thanks, Mark. And to the badass group of women that have taken over the lab since then, you guys are incredible. And thanks for taking me out, tagging, and helping me stick surfboards in your sharks' mouths. I really appreciate it. And Carl, honestly, it's been a treat working with you. I would do it again in a heartbeat, so keep that in mind. I gotta thank my 2014 cohort. A lot of these people became super close friends. We all went through this program together. It's been an honor seeing you guys go off and do amazing things. These two especially, Mara and Beth, who were so encouraging on me applying for my Knaus Fellowship, which is what I'm going on to do after this. Like, thanks so much for the support, the encouragement. I really would not be doing what I'm doing today if I wasn't following you two awesome role models down the policy path. So thank you so much. I gotta thank my family. I've got the best family, you guys. I've got 3 sisters and a brother, an awesome stepdad. You guys are so supportive and there for me all the time. I got to thank my dad as well, who got me out into the woods fishing and hunting when I was little, getting me interested in the natural world. So I appreciate that a lot. And more importantly, my mom and my sister Heather are the 2 people that shaped me the most in my life. My mom taught me to be strong and stand up for myself. And she also instilled in me that me being dyslexic was no excuse for not being good at school, that I was just going to have to work harder. And so that's what I did. I certainly wouldn't be here without that encouragement. And my sister, who shaped who I am so much and teaching me so many things about the world and being such a great role model. I love you two so much, and I'm excited to see you soon. Whew. I'm gonna take a minute. To my super close Hawaii family, Josh, Charlotte, you guys keep me grounded when I'm getting a little out of hand. I love you. I was so stoked to be a part of your wedding. Sean is actually here with me today. He's one of my best friends. We lived together a long time ago. We're about to celebrate our 7th Christmas together. I'm looking forward to that. I fucking love you, man. Leon, you're one of my first friends in Hawaii and still one of the best. I love hanging out with you. You're always a good time. You have such a good heart. Morgan, thanks for keeping me healthy, body and mind. I love your yoga classes. I love our deep talks about life and how we feel about the world. Mariana and Shreya, just thanks for gossiping with me about everything. I love talking to you guys. I have way more friends here than I have time to thank you all. I made this slide super fast, definitely today. So a bunch of you didn't make it. It's not because I don't love you, I do. But my sailing community and my kiting community who have given me just the best recreation I could possibly ask for here in Hawaii. I really appreciate all of you. I got to super thank Sammy for making me an awesome kiter and for keeping me on fresh kite gear all these years. You've been great to work with. I appreciate you and Kim so much. I got to thank all my friends and support group I have back in Oregon. I'm really excited to see you guys all soon. You guys were such a big part of me even making it to Hawaii. And by the time I got here, that was all from your guys' help and support. So thank you so much. One last person. Most of you might not even know who this guy is. This is Mark Marks. He did a lot of research on white sharks and shot for National Geographic. I met Mark when I was distilling. I was getting pretty comfy in the booze industry. It was a fun job. I really enjoyed it, but I knew I always wanted to work on sharks. I eventually met Mark. He told me about his PhD. He coached me on how to get into grad school. I can't tell you how many project proposals he actually went over and how many letters to advisors he went over. So Mark, you're a huge reason I'm here today. I love you. I appreciate you, and I'll see you in a little while. Holy shit. And with that, I think I can take some questions.
Woo!
Yay!
We have time for a few questions. You can use the audio or you can put them in the chat.
I have a question. I'm Derek's mom.
Hi, Mom.
Derek, on one of your first graphs that you showed, and I think it was the bluefin tuna, and you showed the decline in the numbers, and then they went up in the early 90s, and then drastically back down. So what led to that uptick in their population, and then the big decrease again?
What allowed them to recover?
Yes.
I don't actually know in that specific situation. I imagine it might have been some sort of, it could have been some restrictions put in place, or environmentally, they might have just had a really productive year. It was favorable for their spawning. I don't know. It could be a lot of things. But that's a good question. Thanks, Mom.
Yeah, thanks. Derek, a question, but first a point. Most Pacific tunas are sustainably fished. So when you make the statement that maybe they're not, you need to put that in context and document it. And frankly, it's not a true statement. But I'm curious, you noted that in your research, you found that there was genetic heterogeneity in the silky shark population. So there were separate stocks of silky sharks in the Pacific, and suggested that, separate stock assessment should be conducted for those components. As a person who does population dynamics analyses, I ask, from a genetic perspective, how do we draw the boundaries to do those stock assessments? How do we separate the data out? So that's my question to you.
That's a great question. And that was one that came up in my practice talk. So the type of genomics that we did We pooled all of our samples before sequencing. So the resolution we have is kind of the average placement from where we pulled our actual samples, which I have a slide of. Am I still on slide view for you guys? Are you guys still looking at my slides?
Yeah.
Oh,
Yes. Looks good to me.
Yes.
I don't have it on there. I meant to. So I can't actually draw the lines from my research. I would like to be able to. And to do that, what we would have to do is sequence individuals because we would know where they come from. But what would help even more is to sequence young of the year. So if we could get sharks below a certain size or still have an umbilical scar that haven't had the opportunity to swim long distances yet, that would be a really good way to find out a little more precisely where the stock boundaries are. And we weren't able to do that in our analysis, but that would be a way to go about that in the future.
Thank you.
And if you guys are gonna ask a question, I'd love it. Maybe just tell me who it is first, because there's a lot of windows for me to scroll through to find you.
Hey, Derek, this is Kim Holland.
Hey, Kim.
I know I'm going to get a shot at you as a committee member, but I just wanted to follow up on the previous comment. You have to be much more careful about saying what is sustainable in terms of tuna fisheries. And as for somebody that spent their early career trying to identify one species from another, in this case sharks, you're also going to have to be very careful about tunas and which species you're talking about, because some species like the skipjacks are definitely fished sustainably. So I also took a little bit of offense when in then boxing, they'd say you lead with your chin. If you're going to put your chin out like that, you can expect to get a punch on it. So anyway, otherwise, I think you did a really good job.
Thanks. Yeah, maybe I should have been a little more clear that I was using tuna as a general example. I know I said bluefin tuna a lot because there's a lot of data on them. So my apologies. Thanks, Kim.
Derek, it's Michael. I have a question, which I think maybe you've been asked before in other versions of this talk, but I'm interested to hear because I don't remember your answer. When you say some populations are significantly different, and yet they still have fairly low FSTs, where do you draw meaning there in terms of the actual value of the FST versus the significance of the FST?
Got you. Yeah. So that's a question that we've been kicking around the lab a lot, right? So we have low FST values, but because of the power of our data, at what point is a low FST value could possibly come out significant, but actually not have biological meaning? And that's something we're going to test further. I wasn't able to do it in the time of my dissertation, but when we're doing these types of libraries in the future, we hope to do repetitive libraries. So have the same samples, split that in half, sequence both separately. And when we compare those numbers later, theoretically they should be 0. But there could just be some noise in the data to where they will actually have an FST value. But the idea being, if you do that replicate for maybe 3 or more libraries, then you have this kind of baseline noise and you can draw the line there of what a low FST value should be. That help?
Yes, thank you. And I should say that was a really great talk. Thank you. I should have led with that too.
Thanks.
Hey, Derek, I have a question for you.
Okay.
It's Austin Green.
Hey, Austin.
Hey, awesome job, by the way, Michael. Yeah, totally right on. Just from the spit swabs, do you guys have any plans, or I don't know if the spit swabs are consumed in the process, any plans to look at hormones or divergence between sexes of where the bites are coming from?
I am starting a new job in February, so I have no plans of that. However, we still have those samples if you're keen.
Not sure I have the right skill set, but it would just be interesting to know if there's any sort of, especially with the Australia data set, to know if there's any sort of seasonality in behavior with like hormone levels or anything like that.
Yeah, I don't know. I don't know enough about hormones to know if they're going to be present in saliva or not.
Yeah, yeah. Cool. Well, either way, great talk. Thanks so much.
Thanks, Austin.
Hey, Derek, it's Mark Royer.
Hey, Mark.
Hey, great talk.
Thank you.
So I got a good question about the spit swab. Sorry if I missed this, if you did mention this in your talk, but what is the time frame that you can look at for when a bite happens on a person or on an object that you can still get reliable DNA on it? Probably like one of the questions is like, could a bite victim be in a hospital bed and then they can, because the hospitals, doesn't have to go through all the mumbo jumbo of handling a bite victim, if they could do the swab on their end. And also like in a forensics point where sometimes bodies that are lost at sea are scavenged by animals, namely tiger sharks in area around Hawaii. If a shark bit a person, 12 hours, like a whole day before,
Sure.
would it still have viable DNA to do the spit swabbing?
Yeah, we get this question a lot. Our fishermen in Guam and Saipan really wanted an answer. They're like, well, we're fishing, the bite's good. I need to do it like right away. And we'd say, no, do it when you get to it. Well, we don't have an answer. We had set up a project for that. And then due to circumstances, we weren't able to go do our field work and actually test it. But the idea would be we would go out and have some sharks bite a bunch of boards, take a swab right away, set them in the sun, wait 6 hours, take another one, wait a day, take another one, wait 2 days, take another one. The only thing I can comment on that is the engineering department came to me a while ago and they said they had these mooring lines that kept breaking, and they kept breaking at depth and they look kind of sheared, and they're like, we think sharks are biting these lines and like our buoys are coming off. We heard you swab stuff, like, do you want to try it? And I was like, really suspicious, but I was like, sure, I'll give it a try. And their first sample was like weeks old and didn't work. Their second one, they got it within 2 days. So it was in the water for 2 days, and they got it into a freezer pretty quickly, and I was able to get DNA off of it. I tried 2 more that they had recently and didn't have enough time to really try it all the way. So I'm gonna call those 2 inconclusive. But at least that one was in the water and it was still viable after a little while. But being in the water compared to being out in the sun or being in a shaded carport, I don't know. We want to test that, but I don't have an answer.
So for
Very
the
cool,
one
thank
got
you.
The sample off of, the one you got the sample off, Jeff, what was it?
It was a tiger shark.
It was, okay.
Anybody else? Going once?
All right. Well, if there's no more questions, I'll ask our audience to shut down and the committee to stick around, please.
Good job.
Yeah, dude. That's Derek.
Good job, Derek. We love you, Derek. Good job.
We love you, Derek.
Cheers.
Hey, Lalea was watching.
Dr. Kraft, which scotch goes best with the shark talk?
Whichever one's in your hand, Mitch. All right, y'all. You got to get out of here. I'll see you later.
All right.
Good work.
Yeah. Thanks, man. See you soon.
Hey,
Thanks, Sophia.
congratulations.
Thanks.
Congrats. Back.
Hi, Sophia.
Thanks, man. Yeah. Yeah? I'm about to talk, bud. You gotta go. Okay. Oh, my bad.
So Brian, one of us can remove these other people if you want.
Yeah, kick the bums out.
Kick them out. That got their attention.
Sorry, folks, committee members only. Nice to see you, John.
Remove.
Am I still screen sharing? Probably am, huh?
So John Brodziak is not on the committee, correct?
Right.
Okay. And I just wanted to stay on to make sure that as I leave and stop recording, things don't get confused. So Brian, I believe you are one of you, either Derek or Brian, you are host. And so as co-host, I should be able to stop the recording without messing you guys up. In which case, good luck, Derek. Love the talk.